Welcome to Bio 205 Microbiology at Pima Community College. In this video, we're going to show you how to take an inoculation from your skin and your nose to examine different staphylococci that naturally resides in those niches. So what we're going to need for this particular lab is what's called a mannitol salt agar Petri plate, MSA for short. I'll show you what this does soon. 2 swabs, one for your skin, one for your nose. Then we have a test tube here of just some sterile water. This is going to help us pick up some bacteria from our skin. So like always, we're going to label our Petri plate. I'm going to put my initials, today's date, MSA. And then I'm going to divide the plate down the center here. You could use a ruler if you wanted. And I'm going to indicate on one side skin, the other side nose. So to get your skin sample, I'm going to take a sterile cotton swab applicator here. I want to make sure not to touch it to anything else. These can go in the trash. I'm going to dip it just to moisten it in my sterile water. Just for demonstration purposes, I'm just going to lift my coat here. I'm just going to pick a spot on my skin that you guys can see. And I'm going to roll the swab back and forth. See what I can pick up. Inoculation, it's super simple. I'm going to put my finger down on the skin side. This is the part of the Petri plate I want to inoculate. And you can just do, in an S-like motion, an S streak down the plate here. When you're done with this swab, dispose of it. And then we're going to get to check out 24 to 48 hours later what was growing on my skin. The next one is the nose. I don't have to moisten the swab because my nose should be pretty moist. Don't hurt yourself. You're going to take your swab, pick a nostril, any nostril. You can kind of swab the inside. A lot of proper swab inoculations go pretty far back. I don't want to hurt myself. Hopefully there's no boogers on here. Take your swab, go to your nose side, rub it down. Just like we did. Make sure you're not kind of swabbing back and forth from your skin. You don't want to cross-contaminate those 2 samples. Dispose of your swab. We're going to see that certain types of staphylococci are actually harbored in the nose. They can end up on your skin when you sneeze, things like this. But we're going to examine 2 potential species of Staphylococcus that we can find growing, either in the nose or on the skin itself. So once you've done that, that should be super simple and fast. Incubate this, and then we'll come back on day 2 and look at our results. So part 2, or day 2 of the skin lab, I just wanted to remind you guys that this is the original color of the mannitol salt agar Petri plate that we started with. Now, after you inoculate this, and grow it 24 to 48 hours, you have 2 potential results that you may see. And so these are just demos. These are, obviously not the grown cultures from my particular nose and skin that I showed you earlier. But what we have here is the 2 different possible species, 2 of them anyway, that we could see growing on our skin or from our nose. So this first one, Staphococcus Epidermidis, you guys will notice is almost fairly the same color as the original color of the Petri plate started out here, okay? But notice when we inoculate known Staphylococcus aureus onto this type of Petri plate, it turns bright yellow. So what this Petri plate does is it allows bacteria that ferment mannitol into acid to appear as a yellow color. So when comparing and contrasting these two species, Staph epidermidis, think the epidermis, it grows on your skin. It does not ferment mannitol and therefore does not turn yellow. Staph aureus, which can be harbored in the nose of some individuals, will ferment mannitol and turn yellow. So I can differentiate or tell the difference between two species based off of mannitol fermentation. This is my first clue that perhaps I harbor or carry Staphylococcus aureus in my nose. Now, it can end up on your skin again if you were to sneeze, for example. But if you carry it somewhere on your body, it's going to, generally speaking, appear yellow on MSA. We can't confirm a species just by looking at MSA alone. So in the next portion of this video, I'm going to show you two tests that we use to narrow down Staph aureus specifically. All right, so in order to further differentiate Staphylococcus aureus from Staphococcus epidermidis, we're going to perform two tests. The first one is called Catalase Testing, and that's going to involve hydrogen peroxide here. And this test alone doesn't technically differentiate these two species from each other, but sometimes what can happen when we take samples from humans, remember Staph aureus appeared yellow on mannitol salt agar, right? Sometimes you can get mannitol fermenting streptococci. And the difference between these groups is that staphs have catalase and will bubble. Streptococci do not. So whatever's growing here, if we didn't know, we would first test catalase and see, does it fit into the staph group? If so, it'll have catalase. If it doesn't have catalase, it could be streptococci as a potential culprit. So let's see if these two species do indeed have the catalase enzyme. I'm going to flip these over. So we have Staph aureus here, and we have Staph epidermidis. So I'm going to start with Staph aureus first. I'm going to take off my lid. Pick a section. You could do a single colony. I'm just going to pick a really heavily grown area here, drop a little bit of hydrogen peroxide, and that bubbles really nicely. So known Staph aureus certainly does have catalase, so it should bubble if you were taking this from your MSA plate. Let's see if Staph epi can also test positive for catalase. Take a heavily grown area, and sure enough, Staph epi is also positive for catalase. So the Staphylococci, again, are catalase positive. If you had streps growing on your skin and that's what you isolated, they would not turn positive for catalase. Okay, this third part is now really going to help us narrow down. We know that we have some sort of staphs because they're catalase positive, and we want to now differentiate aureus from epidermidis to see if you're a potential carrier for Staph aureus here. So after you incubated your plate, remember we essentially swabbed our nose and our skin. So let's say it came back and we were looking at a result. Start with the Catalase test. We know it's positive. And now we would take this one step further and do what's called Coagulase testing. Many strains of Staph aureus have what's called the Coagulase enzyme. And to test for this, think coagulation or clumping. We're going to test the bacteria from the Petri plates here to see if they have this particular enzyme. And Staph aureus should be positive for coagulase, Staph epidermidis should be negative. So this is how we're really going to further break them down here. So we have a latex reagent. It's going to essentially be coagulated or clumped by the coagulase enzyme. And then we have a little card here. It's very difficult to see, but I've labeled it SA for Staph aureus, and I've labeled this SE for Staph epi, because I want to show you what a positive and a negative result for coagulation would look like. We also have two toothpicks. So I'm going to go from my Staph aureus plate here and scrape up a visible chunk of cells, kind of the more the merrier. You could do a single colony. I'm taking more here just for demo purposes. I don't know if you guys can see the chunk on the end. See, but I have a little mass of cells. And I'm going to rub that right into the center, pause for a moment. I'm going to do the next plate here. Hold that thought. This is known staph epi, which should be negative for coagulase. I have it labeled SE here. Now, we just want to make sure we don't cross-contaminate. Take our latex reagent. This can be clumped by coagulase. But if he drops down, so I'm going to do this one at a time, just so you guys can see this, but you're going to take your toothpick here, and you're going to try and break up any chunks of cells. You're going to mix it here with your toothpick. We're really going to have to zoom in on this. And this is happening quite quickly. You want to use bacteria that are relatively young. You don't want to use old cultures. And you can actually see that clumping is starting to form. Now let's kind of compare that to staph epi over here that should not have any clumping. Hopefully we can see the difference. So I'm mixing in the bacteria, I'm getting rid of any chunks. And besides chunks of cells, I'm not seeing coagulation. I'm not seeing a clumping reaction. So if you guys were to look at this, on the Staph Aureus sample, you can see chunks, coagulation. And if you notice staph epi on this side over here is quite milky, and there's no coagulation occurring. So Staph Aureus, oftentimes, depending on the strain, but most of these are coagulase positive, Staph epidermidis is negative. So between mannitol cell agar catalase testing and coagulase testing, we can really narrow down whether or not someone's carrying Staphylococcus aureus.